Hello, Physiology Lab students. Welcome to the lab. Today's lab, we're going to be looking at osmosis and diffusion, and we're going to look at it doing two simple experiments that last only about 30 minutes. Okay, so what I'm going to do is I'm going to show you how to set up the two experiments, but first, let's look to the online handout for today's lab. Okay, I'm going to share the screen with you. All right. Here we go. Okay, so let's move some of this. There we go. So what we're doing first is we're going to look at osmosis across an egg cell membrane. Now what I've done is I have decalcified some eggs. Here we go. I've decalcified some eggs, which means I soaked them in vinegar, plain vinegar, for 2 days, and the mineral shell dissolved away, revealing a very large egg cell membrane. Now, you can think of this as a red blood cell or any cell in your body, but let's think of it as a red blood cell. Now, if you put a cell in a solution that's very concentrated with salt or even sugar, what's going to happen is water is going to move by osmosis from one side of the membrane to the other, and it will cross the membrane to a side that has more concentrated solutes in it. So if I put that egg in a beaker or a cup of a very salty solution or a very sugary solution, that solution in that cup is considered hypertonic to the contents of the egg. And what water will do is it'll move to the side of the membrane that has more solutes divided. So if I put an egg in a cup of sugar water, that water is hypertonic with sugar compared to the egg. Water will leave the egg by osmosis and enter the cup water to dilute all those sugars. And what should happen is that egg should shrink. Now, conversely, if you put an egg in a plain cup of water that's got nothing dissolved in it, now that water is considered hypotonic to the contents of the egg. So now water will enter the egg by crossing its membrane, and that egg will swell up with water. Okay, now that's what would happen in the human body if a red blood cell was placed in a salty solution or pure water. But we don't have any human cells to work with, so we've got egg cells. So what I've done is I'm soaking one of those decalcified eggs in hypertonic corn syrup. It's a very concentrated, very thick solution of corn syrup. So it's a very sugary solution. All right, so what I'm going to do is when I first worked with the egg, before I put it in the corn syrup, I put it in a cup, and I'm going to make this screen go small. Okay, so I don't know how well that can be read. It looks like it's, there we go. It is kind of backwards from my viewpoint, but what it is, I took the original egg length, and that original length is 16.5 centimeters. And I took its original width, which is 15 centimeters, okay? And so that is the original egg after I decalcified it in vinegar, and then I measured these, and then I put it in corn syrup, and it's been soaking in there for 2 hours. Now, how do you find an egg's length and width? Well, actually, it's quite simple. Let's take an egg here, and let's take a tape measure. All right, here we have a tape measure that measures in centimeters. So all I did is you measure the length from top to bottom, from pole to pole, North Pole to South Pole, if you want to think of it that way. And gently I wrap that tape measure around it, and I got 16.5 centimeters in length. Okay, just like that. Just like that. All right, and so I wrote that down. And then to get the width, it's basically the waist of the egg, okay? And when I look at that number was, what was it again? It was 15 centimeters. So that's how you measure an egg, okay? So that's what I did. All right, so the first thing I must do before I do any additional measurements after the egg is soaked in corn syrup is I want to know what the original measurements were so that I can calculate the original egg volume. So in your handout, there is a formula for it. Check your handout. but basically it's there. Okay, now it's all backwards. I'm looking at it right now, it's backwards. But basically, let me share the screen and I'll show you the formula. Okay, go back to our handout. There it is. The volume of the egg equals a constant of 0.496 times, in parentheses, the egg length multiplied by the egg width squared. Now here's the trick. Before I plug in these numbers that I have on this cup, I told you the original egg length was 16.5 centimeters and the original width was 15. Okay, so what I have to do before I plug this into this formula right here is I have to divide the L by 2 and divide the W by 2. So when I do that, what I'm going to get, and I need my calculator because I can't do this off the top of my head, my original length was 16.5. So 16.5 divided by 2 should be 8.25. Let's see if that's true. And it sure is. So divide by 2 equals 8.25 centimeters. If I take the original width of 15 and then divide 15 by 2, all right, that should be 7.5, and it is. So if I divide by 2 equals 7.5. Okay, so, all right, it is backwards, I know, but now my length is 8.25 and my width is 7.5. Now I'm going to plug that into my equation. So if I take 7.5, that's the width divided, times, and square it, I now get 56.25. Okay, now when I multiply that by 8.25, the length measurement that had been divided, I get 8.25 equals, I get 464.4. And then when I multiply it by the constant of 0.496, I get 230.2 centimeters cubed. Now it is backwards, okay? But I plugged in the width, I squared it. I took that value, I multiplied it by the L, 7.5, and then I got that product and I multiplied it by the constant of 0.496, and what I ended up with was 230.5. Okay, that is my original egg volume, and that's how you do the calculations. Now, your assignment is I'm going to give you the measurements of the egg after it's soaked in the corn syrup for 2 hours, and you're going to calculate the post syrup egg volume. Okay, though, this is going to be messy, and I'm going to have to walk to there near the lab because I have to wash all the syrup off of this before I get it all over the countertop and my computer. So, all right. So I'm going to walk over to the bench over there, and I'm going to wash the syrup off this egg. I'll be right back. I'm back. All right, I have a clean egg, and now I need to measure it like I showed you. So here's my tape measure. I'm going to take and go from pole to pole around the egg, and it feels like it shrunk. All right, its new length after soaking in the syrup is 15. Let me write that down. 15. Okay, and its new width, here we go, is, 13. All right. Now what I'm going to do that I've got the measurements, I'm going to plop that egg into some water and let it sit there for half an hour. So now let's calculate the new volume of the egg after soaking in syrup. So this is going to be your homework. So I'm telling you that the post-syrup egg, okay, I know it's backwards, but I'm going to tell you what it is. The post-syrup egg, its length after the syrup was 15 centimeters. Its width after soaking in syrup was 13 centimeters. Now remember, what you're going to need to do is divide 15 by 2, okay? And that's the L that you're going to put in the formula here. You're going to take the 13, the width, divided by 2, and you're going to put that divided number right here, and you're going to square it. You're going to take the squared W, multiply it by the L, multiply it by the constant of 0.496, and that will give you the volume. So that's going to be your assignment, is to tell me the post-syrup egg volume. Okay, fast forward. It's been 30 minutes. Boom. Okay, that egg has now soaked in water. It was an identical size egg. Okay, it's soaked in the water for about 30 minutes. Woo, that feels plump. Okay, let's dry it off, all right? We're gonna do the same measurements now for the post-water soak. Now, very carefully, because this egg is very filled, I'm going to wrap it from pole to pole and measure, okay? And that is 17 centimeters. So I'm gonna write that down, post-water soak, 17 centimeters. Okay, that does seem like it changed. Okay, now the waist for the width of the egg. Okay, well, that's 15 and a half, 15.5 centimeters. Okay, so your next calculation, your next homework assignment. Post-syrup egg. I know it's backwards. Well, it is for me. The post-water length is 15 centimeters. Nope, grabbed the wrong one. Ha, 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 ha. See, I was testing you. Passed, barely. Okay, this is the water. See the little subscript W? Okay, I knew those numbers sounded bad. After soaking in water, the length was 17 centimeters. After soaking in water, the width was 15 and a half centimeters. Now, you're going to have to plug it into the formula, but before you do, take the 17 and divide it by 2, and then that'll be the length you put in the formula here. Take the 15.5, divide it by 2, and that will be the W you put here. Then you'll square that W, multiply it by the L, multiply it by the constant of 0.496, and that will give you the post-water egg volume. So you're going to have to answer that one for me. All right, so let's look at some class data. Because you weren't in the lab, you're doing the virtual lab. So let me share my screen with you again. Okay, because we had 6 or 7 tables of people complete the experiment. Okay, so here's class data. Now we're just going to make this big so you can just look at the egg experiment right here. Make it even a little bit bigger. Okay, there it is. There's all of our egg volume measurements from the class. So we had 7 eggs that were done. Here we have all the original egg volumes, all the post-syrup volumes, and all the post-water volumes. Let's look at the statistics. So the averages look different. It started at 231, went down to 175. But was that significant? When I compared these 2 data sets with a t-test, I got a t-test p-value of 0.00003. That says that those 2 data sets are very different from each other. And you can say with confidence, the egg did in fact get smaller in volume. Okay, then I compared the post-syrup volumes with the post-water volumes, because what I want to know is when you put the egg in water, did it swell back up again? Okay, you're going to have to find out what happened with your egg. Okay, so when I look at the t-test p-value for that, it's significant. It's 0.037. So it went down to 175.6, but then it significantly increased in volume to 197. And that happened after only 30 minutes of soaking in water. Now for funsies, I compared the original egg volume to the post-water volume to see if maybe the egg swelled up to its original size. but they were different from each other. So the egg did swell after soaking in syrup, but it did not attain its original egg volume size. Okay, so that's the results of our osmosis experiment with the egg. When you put that egg in a hypertonic solution of corn syrup, it shrinks because water leaves the egg by osmosis. When you then take that egg and put it into a hypotonic solution of just plain water, now the water enters the egg, causing that egg to swell up. And that is all happening because of osmosis and tonicity of the solution in which the cell is. Okay, so that's one experiment. We have another experiment going on. In the other experiment, we are looking at osmosis and diffusion across a dialysis membrane, and I'm going to show you how to set that experiment up. Okay, so we're going to stop the share, and we are going to set up that experiment. So come with me. Okay, here we are. So I'm going to get this on the bench. The egg data is done. Okay, now what we're doing is we're going to take a dialysis membrane. Now this is called the dialysis membrane because it's similar to the membrane they use in kidney dialysis. For people whose kidneys have failed, they will basically filter their arterial blood through high quality dialysis membrane, and that's what this is for. Now this membrane has got small pores in it that will allow small things through, but not big things. Now I'm going to come back to that in a second, but the first thing I'm going to do is I'm going to tie a knot in this because I'm going to add some solutions to this bag. Okay, so opening up the other end of the dialysis membrane is always a challenge. So let me take a moment here and voila, I got it open. Now to the inside of this bag, I'm going to add a solution of glucose. Glucose is a simple carbohydrate. It's a monosaccharide. Mono means one and saccharide means sugar. It's a single sugar unit. That makes it kind of small. Okay, I'm going to add 2 pipettes full of this to my dialysis tubing. So I've added glucose, a small sugar. Now I'm going to add 2 pipettes full of starch. Starch is a complex carbohydrate. It's a polysaccharide. Poly means many, and the saccharide refers to sugar units. This has many sugar units in it, and that makes it a very big molecule. Now, remember what I said about this membrane. It has got small pores that will allow small things through, but not large things. Glucose is a small thing. Starch is a big thing. So keep that in mind. Now what I'm going to do is I'm going to put this bag inside this beaker. I'm going to put the open end and hold it in place with a rubber band. I'm going to go to the sink really quickly. I'm going to fill this beaker half full with water. Okay, there we go. Now, to that water, I'm going to be adding iodine. Iodine is an indicator reagent that when it comes in the presence of starch will turn it bluish or black. Okay, so we're going to put enough iodine in the water to turn that water visibly yellow. Okay, and then I'm going to set my timer for 30 minutes, and I'm going to see what happens with osmosis and diffusion across that dialysis membrane. So inside the bag is starch and glucose, a very high concentration of both of them. The beaker water is just plain water. So compared to the contents of the bag, the water is hypotonic. Okay, now the principle of diffusion is particles like glucose and starch want to diffuse with their concentration gradient from where they're in high concentration, like inside this bag, to where there is low concentration on the other side of the membrane, which would be the tap water with iodine. That would be a lower concentration. Right now in that beaker water, there is no starch, there is no glucose, there's nothing but water and iodine. So we expect if something can leave the bag through those small pores, they will diffuse from high concentration inside the bag, outside the bag into the water. Now, I told you that glucose is small. There's a chance, yes, there is, that glucose will leave. Now, starch, being a big molecule, probably will not escape the bag to get into the beaker water. Okay. Time warp. It's been 30 minutes. Here's the experiment. Okay. It's been 30 minutes. Here is our bag. Now, let's just get the water off of it really quickly here. Look at this bag very carefully. Does it look like there's a bunch of blue or blackish flecks at the bottom of that bag? There sure is. Okay. Now, what has happened here? What turned what was previously a milky white solution of glucose and starch blue? Here's the answer. The water was pure tap water at the beginning of the experiment. Now, by osmosis, water wants to cross a membrane to where particles are in high concentration to dilute those particles. So water from the beaker entered the bag by osmosis and carried iodine with it. And so water entered this bag by osmosis, and when that iodine came in contact with the starch, it turned it blue because iodine's an indicator reagent. So we have confirmation that osmosis occurred. Okay, so that's one passive transport. What about the diffusion? Did starch or glucose leave the bag by a diffusion from high to low with their concentration gradient? The answer is, well, how am I going to tell if glucose is in the beaker water? I used a glucose test strip. So I took one of these strips and I swirled it around into the beaker water, thus so, and I gave it a few minutes to sit and react, and then I compared that strip with the code on the bottle. And if it was negative for glucose, it'd be kind of a teal color, okay? It's probably hard to see, but it is certainly not that. That sucker is brown, and that is positive for glucose. So we have confirmed that glucose left this bag. It escaped the pores of the bag because it must have been sufficiently small enough, the glucose, and then it diffused into the beaker water, and the test strip confirmed that it is in fact there. Now, we know starch didn't leave the bag. Well, for one thing, the pores on that bag are not big enough. And two, the beaker water never turned bluish or brownish. If starch had escaped the bag and come in contact with that iodine, this entire beaker would be blue, but it's not. The only thing that's blue is the inside of the bag. Okay, so that's our experiment. Okay, osmosis entered the bag, carrying iodine, reacting with the starch, turning it blue. Glucose escaped the bag by diffusion into the water, and we confirmed its presence with a test strip. So it was a very fun lab on osmosis and diffusion. I hope you enjoyed it. And for my students, I'm going to be sending you a questionnaire based on this video for you to answer. All right. See you next time, Physiology Lab.